In Lab 2, we're going to be learning aseptic technique and how to use that to transfer microorganisms without contamination. We're also going to be learning a lot of terminology related to growing bacteria and other microorganisms. Now, this is another lab where we have pre-lab quiz videos to view. We have a number of them in this lab since it's one of our most important labs of the semester. One dealing with the forms of culture media, aseptic technique tips, how to inoculate broth tubes, slant tubes, and stab tubes using aseptic technique, how to streak Petri plates for isolation using aseptic technique, a video on the procedure for today's lab, and also some photographs showing you isolated colonies of bacteria on a Petri plate. So remember, these videos have to be viewed and you have to complete the Lab 2 pre-quiz found on your LMS site prior to the beginning of that day's lab. Now let me remind you again about the performance objectives at the end of the lab that you have to know for your first lab quiz, which covers the introduction to the lab manual and labs 1 through 4. And the objectives you see under discussion are theoretical objectives, which is basically a lot of terminology we need to know related to growing organisms. You should be able to define pure culture, sterile medium, inoculum, aseptic technique, and colony. State and define the 3 types of growth that may be seen in a broth culture. Define the terms obligate aerobe, microaerophil, obligate anaerobe, aerotolerant anaerobe, and facultative anaerobe, terms that relate to the oxygen requirements of microorganisms. Define the terms psychrophil, mesophil, thermophil, and hypothermophil, terms that relate to the temperature requirements of microorganisms. And then the following terms relating to pigment production, namely chromogenic, water-soluble pigment, and water-insoluble pigment. The objectives under procedure basically say you should be able to transfer organisms aseptically, label everything properly, put it on the proper incubator shelf, return everything where it belongs when you're done, and dispose of all the materials safely as learned in our safety training when you're finished with the lab. And there's only 2 practical questions on this lab since we're mainly practicing aseptic technique and learning terminology. But on the practical for Lab Quiz 1, you should recognize and identify the following types of growth in a broth culture: pellicle, turbidity, sediment, or any combination of these. And finally, state the color and water solubility of pigment seen on a plate of a chromogenic bacterium. And don't forget, at the end of each lab, there's a self-quiz you can do, along with answers for that self-quiz. So let's start out with some of the early terminology, general terms we need to know in order to grow bacteria and to understand aseptic technique. Now, we really need to realize that in natural environments, microorganisms usually exist as mixed populations, a lot of different bacteria all growing together. But if we want to study the organisms in the lab or identify them, we typically have to have the organisms in the form of a pure culture, our first definition. So a pure culture is one in which all organisms are descendants of the same organism. Everything growing in that tube or on that Petri plate or on that slant tube came initially from one microorganism. And we'll learn in Lab 3 how to obtain pure cultures from a mixed population. But in most labs, we'll furnish you with the pure cultures of the bacteria we want to grow. Now, what we need first of all then, if we're going to be growing microorganisms, is a sterile, nutrient-containing medium in which to grow them. So anything we use to grow microorganisms is called a medium. But before we can use it, it has to be a sterile medium. And sterile we define as free of all life forms. And we provide you with that. Typically it's sterilized by autoclaving, where we heat it to a temperature where all contaminating microorganisms are destroyed. And if we're going to work with microorganisms, we have to have a method of transferring the growing organisms from a pure culture to a sterile medium without introducing any unwanted contaminants. So the bacteria that we're going to be transferring, we call the inoculum, and the process is often called inoculation. And the method we use is aseptic technique, and that's simply a method of preventing unwanted microorganisms from gaining an access, aseptic technique. So whether we're transferring bacteria from one tube to another or inserting an IV, changing the dressing on a wound, we're using aseptic technique. We're trying to prevent unwanted microorganisms from gaining access. And keep in mind, of course, in all of our labs, you have to be wearing the correct personal protective equipment. That is lab coat, splash-resistant goggles, and gloves, and appropriate shoes. So part A of the lab deals with aseptic technique. Now, the easiest way to learn that is to watch the pre-lab quiz videos I provided. So the first thing you want to do is look at the videos we mentioned previously, the one on forms of culture media, aseptic technique tips, inoculating broth tubes, slant tubes, and stab tubes using aseptic technique, and streaking a Petri plate for isolation using aseptic technique. So the videos will show you how to do that, and the pre-lab quiz will help you get prepared for that. In the lab manual, we've described all of this step by step, and if you want, you can read through that and look at the illustrations as well. Now we're going to introduce a lot of terminology here that relates to growing microorganisms, and we need to know that for both lab and lecture. First of all, the forms of culture media. The bacteria we have growing are referred to as cultures. And so first of all, we can grow them in tubes containing a liquid medium, such as the trypticase soy broth that we're using in today's lab. And so we call those broth tubes, bacteria growing in a liquid. As we see here, this is a liquid medium with some bacteria growing in it. Now, after incubation, we're going to look for growth. And growth is the development of many cells from a few cells. And there are three forms of growth that we can detect in a broth culture. Now, often it's a combination of two or more of those forms, but we need to be able to recognize those, and that's one of your two practical questions. Recognize growth in a broth culture using the terms pellicle, turbidity, and sediment. So pellicle is where we have a mass of microorganisms floating on top of the broth. And that growth on top is called a pellicle. Figure 7a shows you a bacterium, in this case the Mycobacterium Flei we're going to grow today, forming a pellicle on top. The second growth is called turbidity, which refers to cloudiness. So the organisms appear as a general cloudiness throughout the broth. And figure 7b shows you a nice tube with turbidity. So again, it's not the color we're looking at, but the cloudiness. Notice you can't really see through the tube very well because it's cloudy. The bacteria are blocking light. And the third term we use is sediment. And that's where there's a mass of organisms appearing as a deposit at the bottom of the tube, as we see in figure 7c. So you see this little pellet at the bottom is a sediment. But quite frequently we see a combination of those growth. So notice this tube has both sediment at the bottom, but also exhibits turbidity. So that would be turbidity and sediment. And if we look at the first example, figure 7a, this one, notice, has no turbidity. It's crystal clear. It's not cloudy, but it's clear, but it has a pellicle on top as well as some sediment at the bottom. Now, a second form of culture medium is called a slant tube. And these are tubes containing a nutrient medium plus a solidifying agent called agar-agar. And this is a medium that melts when you heat it, but then solidifies when you allow it to cool. So in this case, the medium has been allowed to solidify at an angle in order to get a flat surface for inoculating bacteria. Figure 8a shows you a slant tube before it's inoculated. And because it was allowed to solidify at an angle, we see this flat surface here that can be inoculated. And in figure 8b, we see a bacterium growing on a slant tube. So we see the growth of the bacteria growing on the flat agar surface of the slant tube. The third form of culture medium is called a stab tube, sometimes called an agar deep. And these are tubes of hardened agar which have been allowed to solidify upright, so we inoculate the tube by stabbing the inoculum into the agar. And figure 9 shows you a stab tube that was inoculated. We're going to use an inoculating loop to stab the stab tubes today, but often a straight wire called an inoculating wire is used rather than a loop. But again, you see the bacteria growing into the agar where it was stabbed. Now we see growth at the top. You always see growth at the top, but that isn't a pellicle because, again, this is not a broth culture. This is a solid agar surface. Bacteria will just naturally grow on top of the stab tube. What we're looking for here is it growing into the stab. And finally, the fourth form of culture medium is the agar plate. And these are sterile Petri plates that are aseptically filled with a melted sterile agar medium and then allowed to solidify. And this gives us a nice wide surface on which to grow and separate microorganisms. And most of the time when we're streaking an agar plate or Petri plate, we're doing what's called streaking for isolation. We're trying to get single isolated colonies of the bacteria somewhere on that plate. And if we take a look at the picture of agar plates here, we see the surface of Petri plate, and notice down here at the bottom of the streaking pattern that you'll be using today, we have some single isolated colonies, not touching other colonies. Now where we began to streak the plate, there's so many bacteria, they all grow together. But if we keep spreading them out and spreading them out, eventually we can physically separate the bacteria on the agar plate so that individual bacteria form individual colonies separated from other colonies. And again, that's an essential technique we need to master in our labs. Now, there are terms that are used to describe single colonies of microorganisms in agar plate, and Appendix A shows you another number of descriptive characteristics along with diagrams, but you don't have to know those terms for your lab. What we do want to realize from this, though, is that different bacteria often produce different looking colonies. We're not going to worry about the terms we use to describe them so much, but rather the concept that different bacteria often produce different looking colonies. And that's important in several ways. In lab 3 coming up, we're going to be given a mixture of bacteria, and we're going to try to get pure cultures of each. And we do that by looking for single colonies of each organism in the mixture, and often those colonies will look different because different bacteria produce different looking colonies. This also allows us to recognize if some airborne contaminants landed on your Petri plate when you're inoculating it, because often the contaminants will look different from the other bacteria you are growing. So those are the 4 forms of culture medium we'll be inoculating today. We're going to inoculate a broth tube, a slant tube, a stab tube, and an agar plate with each of the 4 bacteria we're using for a total of 16 inoculations. 4 bacteria times 4 different media that we're going to grow them on. Now, if we're going to grow microorganisms, we also have to provide the right amount of oxygen. And microorganisms vary greatly in their requirements for gaseous oxygen, but we can place them into one of 5 groups based on their oxygen requirements. An obligate aerobe is an organism that grows only in the presence of oxygen. And they obtain their energy from aerobic respiration, where oxygen is the final electron acceptor. Microaerophils are organisms that require low concentrations of oxygen for growth. They don't usually grow well at higher concentrations, but they do require low concentrations, and they too obtain their energy from aerobic respiration. Obligate anaerobes are organisms that grow only without oxygen, and in fact, oxygen can inhibit or kill them. And they obtain their energy from anaerobic respiration or fermentation. And then finally, aerotolerant anaerobes. Like obligate anaerobes, they cannot use oxygen, but they can tolerate it fairly well. And they get their energy from fermentation. And finally, facultative anaerobes are organisms that grow with or without oxygen, but generally better with oxygen. And they obtain their energies from aerobic respiration or anaerobic respiration and fermentation. And most of the bacteria that we deal with in lab are facultative anaerobes. So those are the oxygen requirements and the terms we use to describe those oxygen requirements, part of our terminology we need to know today. We also have to provide the correct temperature for optimum growth of the organism. Now, microorganisms have a minimum temperature at which they can grow. They have a maximum temperature at which they can grow. And then there's an optimum temperature where they grow best. And we could divide microorganisms into 4 groups based on their oxygen, optimum temperature requirements. So the term psychrophil refers to cold-loving bacteria. Their optimum growth temperature is between -5 degrees Celsius. Remember, 0 is freezing on the Celsius scale, and 15 degrees Celsius. These are often found in the Arctic and Arctic regions and in streams fed by glaciers. Mesophils are bacteria that grow best at moderate temperatures. So their optimum temperature is between 25 degrees Celsius and 45 degrees Celsius. Remember, 37 degrees Celsius would be body temperature. So most bacteria are mesophilic. They grow at moderate temperatures. That includes common soil bacteria and, of course, the bacteria that live in and on our body. Thermophils are heat-loving bacteria. Their optimum growth temperature is between 45 degrees Celsius and 70 degrees Celsius. Remember, on the Celsius scale, 100 degrees Celsius is boiling. These are commonly found growing in hot springs and in compost heaps. And lastly, we have hypothermophils. These bacteria grow at very high temperatures. Now, their optimum growth temperature is between 70 degrees Celsius and 110 degrees Celsius, 10 degrees above boiling on the Celsius scale. These are usually members of the domain Archaea, and they're found growing near hydrothermal vents at great depths in the ocean. So those are the terms we use to describe temperature requirements, which we need to know. And then finally, under theory, a little bit on colony morphology and pigmentation. So another important term we need to know is the term colony. And we define colony as a visible mass of microorganisms growing on an agar surface that usually originates from one single organism or one arrangement of that organism. So a single bacterium, or maybe an arrangement like a streptococcus would be an arrangement forming a colony, or a staphylococcus arrangement forming a colony. Now, different bacteria often produce different looking colonies in terms of their appearance. And again, Appendix A has a bunch of terms that are used to describe colonies, but again, you don't have to know the terms in Appendix A. But keep in mind that different microorganisms do often produce different looking colonies. Now, we need to remember that that colony is a huge mass of microorganisms all growing together that we can see with the naked eye. Many, many microscopic bacteria in sufficient quantity, we can see them with the naked eye. And this is a scanning electron micrograph of a microcolony of Escherichia coli. Now, E. coli is a bacillus-shaped bacterium. And this colony, when it was put under the scanning electron microscope, was about the size of a head of a pin. But when highly magnified, you can actually see the individual bacilli in the colony, the individual E. coli, as we see here. Now, one kind of common characteristic of a bacillus-shaped bacterium is that their colonies tend to have an irregular edge, like we see here. And that's because of the shape of the bacterium and the way they lay down. Because they aren't round, they can't compact as tightly, so we tend to see an irregular edge around the colony. When we're going a coccus-shaped bacterium, often the colonies look more spherical. they have a smooth, round edge because the cocci can compact themselves more tightly together and form a more uniform edge to the colony. And you'll see that on several of the plates. We won't see the individual bacteria, but we can see the shapes of the colony. Now, the most visual characteristic that we can see when bacteria grow on a Petri plate is that of pigmentation or color. Some microorganisms naturally produce a colored compound during their growth. They produce pigment. And so bacteria which produce pigment are said to be chromogenic. They have the ability to produce color when they grow. Now, whether they make that pigment can depend on environmental factors. For example, Serratia marcescens, which we're going to use in several labs, has a deep red pigment at 25 degrees Celsius around room temperature, but doesn't produce pigment at body temperature or 37 degrees Celsius. Now, bacterial pigments can be divided into two basic types, water-insoluble pigment and water-soluble pigment. So let's first take the case of water-insoluble pigment. Most of the bacteria produce water-insoluble pigment if they're chromogenic. And this means that the pigment, the actual colored compound they're producing, does not diffuse out of the organism. It stays inside the bacteria. So as a result, the colonies appear pigmented, but the agar itself doesn't change color. It remains a color of normal agar. Now, when we look for pigment production in bacteria, we need to use a plain medium that doesn't have color. A lot of the media we'll use during the semester has special chemicals added that give it a color. So we can't really detect pigment there because that's the color of the agar, not the color of the bacterium. But on a plain medium then, as we see in figure 10a, with water-insoluble pigment, like this plate of Serratia marcescens, this produces a red water-insoluble pigment. So notice the pigment is staying inside the bacterial growth. When we see single colonies, we see the colonies are pigmented, but the agar itself hasn't become pigmented. It doesn't pick up any color. So the pigment stays inside the bacteria, it doesn't diffuse out into the agar. So that's water insoluble. And when we describe pigment, we're describing two terms, and you have to tell me what color it is and whether that pigment they're producing is water insoluble or water soluble. And again, you want to use the pigment when you're describing it. Water insoluble pigment. Don't say the organism is water insoluble. It's the pigment they're producing that's water insoluble. And not many bacteria produce pigment, so the ability to produce pigment is a pretty important characteristic for identifying that bacterium. Now, on the other hand, if the pigment is water soluble, then the pigment diffuses out of the organism since it's soluble in water and diffuses into the surrounding medium, causing the agar to appear pigmented. And a classic example of that we'll use several times during the semester is Pseudomonas aeruginosa. Figure 10c shows you an early plate of that, which is a brief overnight incubation. Now, this was a plain colored agar. It was tan in color. But notice that this is producing a green pigment, and that pigment is beginning to diffuse into the agar. So we can see the early stages of the water-soluble pigment being produced. As the bacteria grow more and as more and more pigments produced, then it eventually will look like what we see here in figure 10b. So notice that was a plain colored tan medium, but now you see the agar itself has turned green. So the pigment is water soluble. And we'll be giving you a number of demonstrations to look at as part of the results of Lab 2. We're going to be looking at and also growing today Micrococcus Ludwigus. And Micrococcus Ludwigus produces a yellow water-insoluble pigment. So again, we see the color is yellow. That's the first part of describing pigment. But the pigment stays inside the colonies, inside the growth. It doesn't diffuse in the agar, so it's water-insoluble. So on a quiz, you'd say that organism has a yellow water-insoluble pigment. You don't have to know the names of the organisms yet, just be able to describe the pigment it's producing. Another one we're going to grow today is Mycobacterium Flei, and that produces an orange water insoluble pigment. So again, notice that the pigment is orange, but it hasn't diffused into the agar. The agar doesn't pick up the pigment, so again, that would be described as orange pigment, which is water insoluble. We'll also show you a demonstration plate of Serratia marcescens, which produces a red water insoluble pigment. And there's a couple other demos you can look at there as well. Now, Pseudomonas, remember, produces the green water soluble pigment, where the pigment diffuses out into the agar, turning the agar green. So that would be described as a green water soluble pigment. And that is one of the key tests to help us identify Pseudomonas aeruginosa, as we'll see later on in Lab 12 and briefly in Lab 3. So there are 4 different forms of media you're going to use today. You'll be working in pairs, so each pair will need 4 trypticase soy broth tubes. Remember, that's the liquid medium. 4 trypticase soy agar slant tubes that have the slanted surface to inoculate. 4 trypticase soy agar stab tubes where we stab the agar. And eventually 7 trypticase soy agar petri plates, 4 for the 4 bacteria we're growing and 3 extra that we're going to contaminate in step 6. And so each pair will be growing eventually today 4 different bacteria. We're going to grow each bacterium on each of the 4 medium. So we have 4 bacteria times 4 media, that's 16 inoculations that you'll be doing as a pair. But you'll divide up the bacteria, so each of you will be doing 8 inoculations. So the 4 bacteria we're going to use today, we have trypticase soy broth cultures of Bacillus subtilis that we want to grow, broth cultures of E. coli, and broth cultures of Micrococcus Ludwigus. And the 4th bacterium we're going to grow is Mycobacterium Flei, but you're going to take that off of an agar plate rather than out of a broth tube. So you'll learn how to do that as well. So steps 1 through 5 tell you step by step what you're doing today. But we can summarize that in figure 11. So we'll have this diagram up for you to look at today, but that shows you the inoculations you're doing per pair. So the way you might want to look at that is one of you will do these 2 inoculations, all the inoculations for Bacillus subtilis, all the inoculations for Micrococcus Ludwigus, while your partner will use the other 2 bacteria and do all the inoculations for E. coli and all the inoculations for Mycobacterium Flei. So if you're starting with Bacillus subtilis, then you're going to inoculate first a broth tube with Bacillus subtilis, then stab a stab tube with Bacillus subtilis, then inoculate a slant tube with Bacillus subtilis, and finally streak a petri plate for isolation with Bacillus subtilis. So again, each organism is going to go on 4 different forms of medium. Now, while you're doing Bacillus subtilis, your partner might take E. coli and do the same thing. Inoculate a broth tube with E. coli, then a stab tube with E. coli, then a slant tube with E. coli, and finally a petri plate with E. coli. Now, you only want to take 2 bacteria at a time, one for you and one for your partner, because we have to share cultures. So as soon as you finish with the first 2, then return them and pick up the other 2 bacteria. So one of you will inoculate a broth tube with Micrococcus Ludwigus, then a stab tube with Micrococcus Ludwigus, a slant tube with Micrococcus Ludwigus, and streak a petri plate for isolation with Micrococcus Ludwigus. While your partner will take the plate of Mycobacterium Flei, our 4th bacterium, and also inoculate a broth tube, then a stab tube, then a slant tube, then a petri plate for isolation. So that's going to give you 12 tubes to inoculate per pair and 4 petri plates to inoculate per pair. And remember to return the class cultures so other classes can use them. Don't incubate the class cultures because other sections of your class will need to use those same cultures for their lab. And all of those bacteria that you inoculate with the pure cultures are going to be incubated at 37 degrees Celsius on the incubator shelf that corresponds to your lab section. So the tubes that you inoculated will be incubated in your test tube rack, and then the test tube rack will be put at 37 degrees Celsius. Now remember, when you start the lab, before you do inoculations, make sure you label every broth tube, stab tube, slant tube, and Petri plate with the organism you're growing on that plate. BS for Bacillus subtilis, EC for E. coli, ML for Micrococcus Ludwigus, MP for Mycobacterium Flei. Make sure you have them labeled so you know which organism is growing in which medium next time. And then the 4 Petri plates that you're inoculating, you're going to put in your Petri plate holder and incubate that also at 37 degrees Celsius on your incubator shelf that corresponds to your lab section. So again, for steps 1 through 5, you'll have 12 tubes and 4 Petri plates per pair to inoculate. But to show that microorganisms are everywhere and that it's always raining bacteria, you're going to take 3 extra plates of trypticase soy agar and you're going to purposefully contaminate them. And you're going to do this as shown in steps 6A, 6B, and 6C. So to start out, once you pick up your materials, first remove the lid of your first contaminant plate and place the exposed agar portion. We're going to go inside the building since we can't go out wearing our lab coats, and leave it out the whole lab. So you want to do this plate first once you get ready to start, so the plate can sit open for the entire lab so that airborne microorganisms can fall onto the agar and show you airborne contaminants that can get into your tubes or onto your plates if you don't use proper aseptic technique. Now at the end of the lab, put the lid back on your air contamination plate, label that plate air so you know that's when you expose to the air. Incubate that upside down, but notice this is going to go in the room temperature incubator or 25 degrees Celsius incubator rather than at the 37 degrees Celsius incubator. So 2 plates only today are going to go at room temperature, the air plate, and then the next plate in step B, the swab plate. Everything else today will go at 37 degrees Celsius. So make sure you double check your incubation temperatures so you get the air plate and the swab plate in the room temperature incubator. And again, we always incubate our plates upside down, as we learned when we did part of our safety training. The second contamination plate you can do later in the lab. Take a wax marker and divide a second contamination plate in half, a sterile plate. You and your partner each get a swab and moisten it in sterile water, and then rub some surface somewhere. And using that swab, streak that all over your half of the second agar plate. So you have half the plate for your swab and the location you chose to streak or to culture, and your partner gets the other half of the plate for their swab. Now this plate will also be incubated upside down at room temperature. That's the second plate today that goes at room temperature. Make sure you label your half the plate with the source of where you got the swab for the organism. So for example, one popular source is to rub the bottom of your shoe. And of course, soil is mainly microorganisms recycling nutrients. So obviously there's a lot of bacteria and molds, spores that are found on the bottom of your shoe. And this is a typical shoe plate shown here with all the different organisms. And you notice many different looking colonies. So each colony represents one organism that fell on the plate, but each different looking colony would be a different organism. By the way, here's a typical air plate, the one that's exposed to the air. And that's stuff that dropped on the Petri plate while the Petri plate was open, and that's the stuff that can drop in and contaminate your cultures if you don't use proper aseptic technique. Remember, it's always raining bacteria. So you have airborne bacteria dropping in from dust particles. You have bacteria from your body dropping in if you aren't careful. So again, each organism, each colony would be one organism that fell on the plate. Each different looking colony is a different organism. Now on the contaminant plates, we'll probably see a number of molds. We can recognize molds because molds look kind of fuzzy when they grow. These are mold cultures here. This is a mold colony. This is a mold colony. Whereas these are probably bacterial colonies. because they don't look fuzzy. They're not composed of hyphae, as we'll see molds are, but rather of microscopic bacteria. And in the final contamination plate, you're going to take a wax marker and divide that plate into quarters and label it according to figure 12. So the 2 quarters on top are labeled glove, the 2 quarters on the bottom are labeled fingers. Now this will be the last plate you do. Before you do this plate, make sure you've put everything else that you've inoculated into the incubator already and that you returned all the class cultures, because you're going to remove one of your gloves here during the process of doing this plate. So you want to be done handling the microorganisms before you do your fingers plate, as we call it. So before you do anything, you'll take your quarter of the plate and rub the fingers from your gloved hand over the surface of this. Your partner will take their gloved hand and rub their fingers over this surface. Now, after you've done that, remove one of the gloves. Remember to put the glove in the biohazard container, not in the trash can. And then rub your fingers over your finger quadrant here. And your partner, after he or she removes his or her glove, will streak their fingers on this sector. And that will show you a lot of the microbiota that live on the fingers. The bacteria constantly growing there. Then at that point, take your gloved hand that you still have the glove on and take that plate back to the 37-degree incubator and put it in your Petri plate holder along with your other plates. At that point, then, you can remove the other glove, wash and sanitize your hands. Now, next time we'll be looking at the results. So we're going to look at our 4 broth tubes, the bacteria growing in liquid, and then you're going to describe the growth you see for each of the 4 bacteria using the terms pellicle, turbidity, and sediment. For example, the E. coli here has turbidity and sediment. Then you're going to observe the growth on your 4 slant tubes. And here's a picture of the 4 bacteria growing on slant tubes. Now, you don't have to draw these. You don't have to know anything about them. You just want to check the 4 tubes and make sure the bacteria grew. If they didn't grow, then you obviously did something wrong. You sterilized the loop after you put the bacteria on, or you did something wrong in the process, because obviously the bacteria should grow on the agar. So again, just observe your slant tubes, make sure all bacteria grew. And the same thing with your 4 stab tubes. There's nothing you have to know on the stab tubes, nothing you have to draw, nothing you have to know for a quiz. Just check the 4 stab tubes, make sure the bacteria grew. We're just doing those for practice because you will be inoculating a couple of stab tubes during the semester. And finally, in step 3, we're going to look at a single colony of each of the 4 bacteria that you grew. So you want to look on the Petri plate for a single isolated colony. Now, step 3, it says using the terms of Appendix A. We're not going to do that. So you can cross out this whole table under step 3. You're not going to fill that in. But what I do want you to do, and we'll be doing this in class, is to look at each of the 4 organisms so you see that the colonies do look differently. We're not going to use the official terms that are used or anything like that, but we want to see they look different. For example, here's a single colony of Bacillus subtilis on an agar plate. And again, we can see here a nice 2 single colonies. We can see that it has an irregular edge. In fact, Bacillus is named after the fact that it is a bacillus-shaped. And remember, as we said, bacillus-shaped bacteria often have an irregular edge just because of the shape of the bacteria and the way they lay themselves down as they replicate. It kind of raises up in the center. When you look at the plate, by removing the lid and looking at it, you'll see the colony looks very dry. And you'll see that these will be very large colonies compared to the other bacteria. So just by looking at them, we can see it's going to look different than the other 3. Here's a single colony of Micrococcus Ludwigus. Now this is a coccus, so they compact better. So rather than an irregular edge, we see a nice smooth edge around the end because they can compact more tightly together as they grow. It's a nice circular and convex colony. And it's kind of moist looking when you look at it from the top. And of course, you see this also produces a yellow water insoluble pigment. If we look at a single colony of E. coli, again, we can see that it has a little bit of an irregular edge to it. Not as obvious as the Bacillus subtilis because this is a much smaller bacterium, but we do see a bit of an irregular edge. We see it has no pigment, just as the Bacillus subtilis had no pigment. The colony is kind of moist looking when you look at it. And of course, it's much smaller than the Bacillus subtilis, as we mentioned. If we look at a single colony of Mycobacterium Flei, this is a bacillus-shaped bacterium. It has an irregular edge to it. You'll notice that when you look at the colony with the lid off, that it's very dry looking. And it has an orange water insoluble pigment. So again, we're not going to fill out the table for step 3, but we do want to observe the 4 bacteria and see that they do produce different looking colonies. You'll also want to observe your 3 contamination plates and note the difference in colony appearances from the different microorganisms that contaminated that plate. And finally, you're going to look at the demonstration plates of several chromogenic bacterium and write down the color and the solubility of the pigment. We're going to look at Micrococcus Ludwigus that has the yellow water insoluble pigment, Mycobacterium Flei that has the orange water insoluble pigment, 2 of the bacteria you're using today. And we're going to look at a plate of Serratia marcescens that has the red water insoluble pigment. And finally, we'll look at a demonstration of Pseudomonas aeruginosa that has the green water soluble pigment. And there's a couple others you can look at as well if you want to practice recognizing color and solubility of pigment. So that takes care of the voiceover for Lab 2, aseptic technique and terms we use when describing growth of microorganisms.